Hello, my name is Amber Russell. I have been studying in the lab of Dr. Derrick Brazill at Hunter College, and my project was to investigate whether PkcA plays a role in the regulation of cell adhesion sites. So first off, it's important to know the function of cell adhesion. Cell adhesion plays a role in many biological processes like organ formation, organ structure, and immune function. Here I've attached a video that demonstrates the use of adhesion in regard to immune function. Here you can see white blood cells moving towards and attacking a parasite. In order for the cells to attack the parasite, the white blood cells must be able to adhere to it. And by the end of the video, you can see the hundreds of thousands of white blood cells attached to it. When there are abnormalities in the cell's ability to adhere, that can lead to various diseases, an example being the metastasis of cancer or the spread of cancer. Now, in order for proper cell adhesion to occur, it needs to be regulated, and this is done through signal transduction. So when signal transduction occurs, it activates actin, alerting it to change its distribution, giving the cell its shape. Example here, I have pictures of platelets, and the platelets are responsible for wound healing. So say you have a cut, you need these cells to get to the wound site. The first picture are how the platelets are before signal transduction has occurred. The cells want to be like this with no real actin structures. Therefore, they have no real function and they are non-adherent. In the second picture, signal transduction has occurred and the actin has been activated, causing the cell to form tendrils that adhere to the wound site and retract to close it shut. The signal transduction is responsible for what has occurred. Signal transduction led to the cell changing its shape and behavior, which led to the generation of adhesion sites, which allows the cell to stick to different surfaces. So what exactly is actin? Actin is a multifunctional protein that forms microfilaments. It can be present as a free monomer G-actin, which forms polymers, which is F-actin. F-actin is organized to perform specific functions, and F-actin organization is controlled by signaling proteins. Actin is also very important in cell motility, cell division, cell signaling, and is an essential cytoskeleton component. Now, in order for actin to be activated and for adherence to occur, a variety of signaling proteins are needed. The specific signaling protein that I'm studying is protein kinase C. Protein kinase C is a signaling protein that phosphorylates other proteins. It is activated in response to other signals, and it regulates a number of different functions in mammalian cells such as cell proliferation, cell migration, and cell differentiation. And all of these things involve adhesion. And if these don't occur properly, this can lead to disorders. So the question is, does PkcA regulate adhesion? The problem is that mammals have about a dozen PkcA proteins, 10 to 15 to be exact. So the solution is to study PkcA regulation in a simpler model, Dictyostelium Discoideum, which from now on I will refer to as Dicty. Dicty is an amoeba-like organism that lives in the soil and eats bacteria and yeast. Adhesion is especially important for them because they need to be able to adhere in order to ingest and capture their food. Moreover, it's a great model because it's genetically simple and has conserved signaling pathways, which means what is found about Dicty's PkcA can be applied to mammalian PkcA. So I mentioned earlier that mammals have about a dozen PkcA proteins. Dicty only has potentially 2, and I'm studying PkcA. PkcA is an ortholog of mammalian PkcA, and loss of PkcA causes defective cell motility, defective cell-cell adhesion, and what I'm studying, defective cell-substrate adhesion. Now, in order for cell-substrate adhesion to occur, the cell has to contain adhesion sites. So what are adhesion sites? Adhesion sites are actin-rich structures located at the interface of the cell membrane and the substrate. Here I have an image to illustrate this. So here's the cell, and at the bottom of the cell are purple dots, which represent the actin-rich adhesion sites. These adhesion sites allow the cell to stick to the surface of the substrate, which brings me to my last key term, cell-substrate adhesion, which simply refers to the cell's ability to stick to a surface. So when the cell lacks these adhesion sites, it affects their ability to adhere to the substrate, which brings me to my hypothesis. Cells lacking PkcA, PkcA- have defective cell substrate adhesion because they have fewer adhesion sites. In order to quantify the number of adhesion sites in the wild-type AX2 cell and the PkcA- cells, I fixed the cells to a chamber slide, stained the actin with phalloidin alexafluor 546, and visualized them using spin disc confocal microscopy. After completing steps 1 through 3, I got this image here. After that, I counted the adhesion sites, and here I pointed out 4, but this cell had a total of 12. After that, I measured the surface area using ImageJ, and ImageJ simply draws an outline around the cell and calculates the surface area from there. So my results were loss of PkcA decreases adhesion sites. On the left, you can see my cells that are stained. The red is the actin that is stained, and the blue is the DAPI that stained the nuclei, and the nuclei was stained for identification purposes. Here you can see that the wild-type cell has 4 adhesion sites, while the PkcA- cell has none. And to avoid bias, I used a large sampling size of 50 cells per the 3 experiments for each cell type. So a question arose while analyzing the data: what if the adhesion sites were just based on cell size? And what if the wild-type cells are bigger than the PkcA- cells, and that's why they have more adhesion sites? So to account for this, I looked at the adhesion sites per surface area, because the amount of adhesion sites between the 2 cell types could have been proportionally the same. The graph on the right shows the results of the adhesion sites per micrometer squared. And when I took the average amount of adhesion sites per surface area over all 3 experiments performed, the AX2 cells consistently had more adhesion sites per surface area. The difference on average was that the wild type had twice as many adhesion sites per surface area, which was found to be statistically significant. So from these results, I can conclude that PkcA- cells have fewer adhesion sites per surface area than the wild-type cells, that the defective adhesion in PkcA- cells may be due to having fewer adhesion sites, which supports my hypothesis, and that PkcA plays a role in the regulation of adhesion sites. A future direction for this project could be to identify the potential targets for PkcA, and we can do this by looking at and comparing the phosphorylated proteins in the wild type and mutant cell, and looking at those different proteins, and from there we can look to see which of those proteins regulate adhesion. And secondly, we can determine how PkcA regulates the formation or destruction of adhesion sites. And to do this, we will look at the formation of adhesion sites in real time in the wild type and mutant cell and see if there's a difference in the formation or destruction time. We started this project in the lab, and here you can see the live cell moving, and you can see the adhesion sites being created and disappearing. And like I said, we would look at the wild type cell and the mutant cell and see if there's a difference between the 2. I would like to thank my mentor, Dr. Derrick Brazill, SPUR, and the National Science Foundation, as well as my fellow lab members. Thank you.